I also received an email from you guys, and then they suggest I go through the study guide. So I thought maybe, yeah, it's a good idea. So I'm not going to go to every question, right? Some questions come pretty easy. So maybe just a couple of questions that I highlighted, thought maybe a little bit complicated. How about that? Okay. But I still prepared the slides, but I will upload the slides in case you guys need it. And I also will use the slide when I get to the questions. So the first part is lipogenesis. I guess you guys are okay, right? For the first 1 to 4 question 1 to 4. And I thought number 5 may be a little bit complicated. But you can let me know if you want me to, go through specific question. So number 5, how does acetyl-CoA get across the inner mitochondrial membrane? Do you guys have a study guide with you? Yeah. Okay. Number 5, all right. Lipogenesis, number 5, question number 5. How does the acetyl-CoA get across the inner mitochondrial membrane? And what is the purpose of this process? Or in other words, what is the role of a citrate shuttle in lipogenesis? All right. So for the lipogenesis, the substrate is the acetyl-CoA, right? Acetyl-CoA is the starting molecules for lipogenesis, the fatty acid synthesis. And we know the acetyl-CoA is generated in the mitochondria. And that needs to be transported out of mitochondria to cytosol because lipogenesis, the whole process occurs in the cytosol, right? So the citrate transport, the purpose of citrate transport is to help the acetyl-CoA get across the mitochondria and to the cytosol for lipogenesis, right? And the specific process you probably want to remember is actually a citrate, not the acetyl-CoA itself, right? Citrate being transported from mitochondria to cytosol and then cleave, back to the acetyl-CoA, right? And don't be confused by acyl-CoA transport into mitochondria, right? And this process is catalyzed by a facility by which enzyme, remember? Yeah, CPT1, right? So don't be confused, the acyl-CoA transported from cytosol into mitochondria for fatty acid oxidation. And number 9. So what is the most important step of lipogenic regulation, and how is lipogenesis regulated allosterically, covalently? The question actually just how lipogenesis is regulated, right, by several different regulators. I have a slide, I guess, number 14. Okay, so this is slide. I'm going to adjust this specific. That's a question number 9. So covalent regulators is actually citrate. Citrate is the positive regulator, right? Can stimulate the lipogenesis. And also, I want you to remember this very important key reaction. The acetyl-CoA is converts to malonyl-CoA, right? So malonyl-CoA formation is the key reaction in the lipogenesis, and this is a key enzyme, right? Acyl-CoA carboxylase, ACC. and regulates this melanocortin formation. And the regulators, all the three type of regulators that we talk about here is regulate this specific enzyme ACC, right? So citrate basically stimulates ACC activities, and long-chain acyl-CoA inhibits ACC activity, right? So that is also the reason why high-fat diet suppress the lipogenesis, because of the long-chain acyl-CoA level will be high when we consume a high-fat diet, right, in the circulation. So this long-chain acyl-CoA can serve as the negative regulators of ACC, so inhibits ACC activity, inhibits lipogenesis. And covalent regulation basically regulates the phosphorylation and dephosphorylation of ACC, and the two hormones, the insulin basically stimulates ACC, right? And glucagon inhibits ACC. And the gene expression, insulin and glucagon can also regulate ACC, but at the gene expression level, and not only activity level. Activity level basically regulates phosphorylation, dephosphorylation, right? And the gene expression just directly regulates the transcription of this enzyme. So this is number 9. And number 12, the glycerol 3-phosphate can be synthesized from which two pathways? Can you guys remember? The two metabolic pathways that very important for the generation of glycerol 3-phosphate. Remember? Glycolysis, right? And the other pathway is directly phosphorylation by the enzyme called hepatic kinase, right? Let me see, that is number 17. So this is a slide for the question number 12. So we have two pathways that can generate glycerol 3-phosphate, glycolysis, or if we also can include in gluconeogenesis, but glycerol glycolysis, I think that's enough if you can remember this, right? So this is the one pathway that for generation of a glycerol 3-phosphate, and the other pathway is the glycerol phosphorylation directly, right, by glycerol kinase. And also question asking which enzymes, right, that involve in the adipose tissue for glycerol 3-phosphorylation or in the liver. So the answer is the glycerol 3-phosphate dehydrogenase is the enzyme that catalyzes formation of glycerol 3-phosphate in the adipose tissue, right, and through the glycolytic pathway. And liver, the glycerol kinase is the enzyme that responsible for the generation glycerol 3-phosphate in the liver, right? So the two pathways in two different tissues and catalyzed by two different enzymes, right? Number 14, where do fatty acids present in the circulation come from? So what are the sources of fatty acids in the fat and fasting condition, right? So this is a slide we talking about under the fat condition, right? We also have a high levels of fatty acids, but this fatty acid mostly from diet, right? And also from liver VLDL, right? Because under the fat condition, liver also does the lipogenesis by convert glucose to fatty acids and be reacidified triglyceride, right, packed into VLDL and released into the circulation. So that also contributes to fatty acids, right? But again, majority of fatty acids under fat condition are from diet, right? So so triglyceride synthesis under the fat condition, of course, the adipose tissue is the main tissue, right? And that very active for the triglyceride synthesis pathway, right? But again, in the intestine, also have a lot of triglyceride synthesized from fatty acids that initially absorbed from the diet, right? And the liver also does the triglyceride synthesis. And under the fasting condition, so the main fatty acids, the most of fatty acids in the circulation are coming from adipose tissue, right? Number 15. So where are the triglycerides synthesized in the fat state, and how are they transported out of these tissues and then to other final destination? I just described, I think, in the last slide. So fat condition, right? Adipose tissue is the primary tissue that synthesizes triglyceride, right? So a lot of triglyceride is synthesized in the adipose side, adipose tissue from fatty acids, which initially uptake from the circulation, right? And also, I said the intestine, small intestine, also have the triglyceride synthesis pathway active, and also in the liver. But again, the most active tissue under fat condition for triglyceride synthesis is adipose tissue, right? And how they are transported out of this tissue. So under the fat condition, we have a lot of triglyceride synthesized in the small intestine and also in the liver, and this two sources of triglyceride eventually all be transported to adipose tissue for energy storage, right, in the form of triglyceride. And number 16, describe the uptake of triglyceride by peripheral tissue. So what is the purpose for taking them up? And what enzymes is involved, and which cells take up fatty acids in the fat condition, again in the fat condition. So what is so which tissue takes up a lot of fatty acids of triglyceride for storage? Adipose tissue, of course, right, under the fat condition. And the purpose of this is to store energy, right, as the triglyceride. And what enzyme involved in the uptake of a triglyceride? LPL, right? Lipoprotein lipase here. Because triglyceride needs to be break down to fatty acids, then be able to taken up by fat cell. So LPL is the enzyme that helps uptake of triglyceride. And 17, so what is the function of LPL, and how is LPL regulated in adipose tissue, muscle, in fat state? I think we have a specific slide, right, talk about the LPL. So LPL, the main function of LPL is to hydrolyze triglyceride in both lipoprotein, two lipoproteins, still remember, chylomicron and VLDL, right? And to break down triglyceride in these two lipoprotein and release fatty acids and for uptake. So this is the main purpose or function of LPL, and LPL again can be synthesized in the two tissues, right? Adipose tissue and muscle, and the regulation of synthesis of LPL in these two tissues are different. So fat condition, adipose tissue LPL is activated and synthesized and released, right? And hydrolyzed again triglyceride in VLDL and chylomicron, and to release fatty acids, and this fatty acids is taken up by adipose cell, and the purpose of this uptake is for synthesis again back to the triglyceride, right? Stored in the adipose tissue. But in the skeletal muscle, the LPL is activated during which physiological condition? Fasting, right? During the fasting, muscle LPL is activated and hydrolyze only VLDL triglyceride, right? Because on the fasting, you wouldn't expect or have a higher levels of chylomicron, right? So only hydrolyze VLDL triglyceride and uptake fatty acids into muscle cells. And what is the final purpose of the fatty acids once it's uptake into the muscle cells? Will be oxidized, right, for the energy. So try to remember this, difference and how they regulate it and which substrate triglyceride, in which lipoproteins, right, LPL hydrolyzed triglyceride, and what is the fate of fatty acids being uptake into the cell. Okay, so I guess that's all the lipogenesis, the question I saw that need to be talked about. What about you guys? Do you have other questions in this specific session that you want me to talk about? Yes? Number 4, okay. Oh, number 6. Describe the 4 main steps that reactions involve making fatty acids. Okay, so I think you probably don't want to remember specific steps. I'm sorry, the question put there, but just try to help you to understand the process. But what you need to know is the rest of the question, like what are the donors of a carbon unit, electron donors for fatty acid synthesis, and also you probably wanted to know the the cycle, right? So each cycle you adding. Let me go back to see it. Okay, so this slide, right? But you don't need to remember exactly, how which enzyme catalyzes condensation or which enzyme, respond for reduction or dehydration. But the kind of 4 steps, right? And most importantly, you need to know this synthesis process is actually cycling, right? So each cycle you're adding 2 carbon unit, right, to extend the 2 carbon. So the 2 carbon unit donor is actually malonyl-CoA, right? And it's also need the NADPH, and you need to know where the NADPH comes from, right? We have 3 sources, right, for the NADPH. One is the pentophosphate pathway, and the second sources of NADH is Maillard enzyme catalyzed reaction, right? And the third is the maybe the next slide. Yeah. So the third sources of NADPH is the isocitrate dehydrogenase reaction. Right? And again, the substrate of the fatty acid synthesis is acetyl-CoA, right? And where does acetyl-CoA comes from for fatty acid synthesis? Three sources, right? Oxidation of glucose, amino acids. Sorry, not three. So I mean the oxidation of glucose and amino acids, right? Okay. Any other questions for lipogenesis? Yeah. Electron donor is NADPH. Okay. Other questions? No. Right. So now let's go to next lipolysis, right? So question 19, describe the process of lipolysis or hydrolysis of triglyceride. Yes, let me see. So this slide, I think, is very important, right? Hydrolysis of triglyceride or lipolysis, we have three different names, right? Called this process, like lipolysis, mobilization of triglyceride, or hydrolysis of triglyceride, right? All the same thing, talking about the same process. So mainly, and this process occurs in the adipose tissue, right? Cell, and the lipase that involved in the hydrolysis of triglyceride, right? Break down triglyceride into three free fatty acids and the glycerol, and this process occurs under the fasting condition. And we have three different lipase that involved in this whole process, right? Hormone-sensitive lipase, ATGL, adipose triglyceride lipase, and mono is glycerol lipase. But I just wanted you to remember the HSL, right, as a key enzyme that catalyzes lipolysis or the mobilization of triglyceride. And HSL is produced inside of cells. It's different from the LPL, right? LPL and HSL basically hydrolyze the same reaction. Catalyze the same reaction, hydrolyze triglyceride, break down triglyceride, but they have a different location, and also these two lipase are regulated differently, right? Based on the physiological condition, like LPL in adipose tissue is activated by insulin during fat condition. Well, actually, HSL is regulated by the other hormones, right, like glucagon, epinephrine, and activated during the fasting condition, right? And one is in the inside of cell, and the other one is in the circulation. And regulation, also I wanted you to know that epinephrine and glucagon is activate HSL, right? Question? No, HSL in the cells, and LPL in the circulation, yes. And epinephrine and glucagon these two hormones basically is upregulate or increase during the fasting, right? And they basically phosphorylates HSL and activate HSL. So increase hydrolysis, right, of triglyceride or increase lipolysis. But insulin basically inhibits HSL, right? And during the fasting, because insulin level is low, so this inhibition will be somehow relieved, right? So that in other words, that insulin, decreased insulin causing increased HSL activity, right? Does that make sense? Okay. So this is for the question number 19. Now 21. Number 21. So does aerobic exercise stimulate lipolysis? Yes or no? Yes, right? Because during exercise, the epinephrine levels increase, so this will stimulate HSL, right, and stimulates the lipolysis. And what hormones play a role in this process? The epinephrine, right? 22nd, 22, number. Okay, does a low carbohydrate diet induce lipolysis? Yes or no? Low carbohydrate diet. Yes, right? So answer is yes, and explanation. Because during the low carbohydrate diet, we have a lower glucose relatively, right, compared to high carbohydrate diet. Have a lower glucose level, blood glucose level, that will kind of mimic the fasting condition. So can stimulate the epinephrine levels increase and decrease insulin level, right? So that will also causing the upregulation of HSL and increase lipolysis. Okay, 24. So how is lipolysis regulated by a hormone? I just described, right? Insulin, glucagon, catecholamine, epinephrine. So again, this slide is very important. 25. What is difference in location, substrate, and activation in response to physiological conditions between HSL and LPL? I also talked about this, I guess, right? Okay. Any other question you want me to address for lipolysis? Yeah, okay. Okay. Now, fatty acid oxidation. 28. So circulating fatty acids are bound to what molecule? Albumin, yes. 29. What is the relationship between fatty acids, oxidation, and plasma free fatty acid level? For example, if we have a higher free fatty acids level in the circulation, what do you expect fatty acid oxidation activity would be? Increase, right? If you still remember, we have a specific slide talk about the regulation, which would be right? Oxidation of fatty acids, regulation of fatty acid oxidation. So basically, plasma levels of fatty acids increase or increase fatty acid oxidation. And the mechanism, I want you to understand the mechanism that will help you to answer the exam question. So, for example, under the fasting condition, right, when we have higher fatty acids that being usually released from fat tissue, right, because of a high HSL activity. So lipolysis increase, so we have more fatty acids being released from fat tissue. So once we have a higher fatty acids level in the circulation, fatty acids can be activated to acyl-CoA. So acyl-CoA levels also increase, which will regulate ACC, right? So basically inhibits ACC activity. And in the meantime, because of we have also glucagon levels increase, so glucagon also inhibits ACC. So in general, when we have higher levels of fatty acids in the circulation, the ACC activity is inhibited, and ACC is the key enzyme in lipogenesis, right? The pathway, and the key reaction for this enzyme that to catalyze is the formation of a malonyl-CoA, and malonyl-CoA is the key substrate you guys have to remember. That is the strong inhibitors of CPT1, right? So once CPT1, and because of, again, under fasting condition, lipogenesis activities decrease, so ACC activities decrease, so we have a lower malonyl-CoA being synthesized, right, level. With lower malonyl-CoA levels, we're causing increased ACC, so which will help transport more fatty acyl-CoA from cytosol to mitochondria for beta oxidation, right? So this is sort of the mechanism, right? So, okay. Now 29. So what is the relationship between fatty acid sorry, I just finished. 32. So how does malonyl-CoA regulate CPT1, right? This slide. How does malonyl-CoA regulate CPT1? Inhibits or stimulates? Inhibits, yes. Okay. And then 38. Are alt-chain fatty acids glucogenic? Do you guys know the answer? Yes or no? Yes, right? Because remember that the fatty acids oxidation usually break down only 2 carbon units at a time, right? We're also doing cycles, fatty acid oxidation, beta oxidation, and each time you generate acetyl-CoA, right? So for the even chain fatty acids, then no problem. All the final product will be acetyl-CoA, right? But the alt-chain fatty acids, the one product, final product, will not be the acetyl-CoA, but the 3 carbons called primary-CoA, right? And if you still remember, primary-CoA is one of the precursors of gluconeogenesis. You still remember, right? So just try to remember the 2 carbon substrate will not be able to use for gluconeogenesis, but 3 carbon unit, yes. Right? So alt-chain fatty acids oxidation, because will generate 3 carbon substrate, right? So that can be used for gluconeogenesis. Okay, 39. How does liver metabolize fatty acids during fasting? Does liver completely oxidize fatty acids through TCA cycle? What do you guys think? Liver? No? But how does liver? Yes, in making ketone bodies, right? So fatty acids go to the liver during the fasting condition, of course, right? They go to the liver, and liver oxidize fatty acids through the beta oxidation and generate acetyl-CoA. And all this acetyl-CoA molecule in the liver during the fasting condition will not be completely oxidized through the TCA cycle, but its acetyl-CoA molecules will be used for synthesis of ketone bodies, right? So it is different from something happening in the muscle, right? Muscle usually will completely oxidize fatty acids for energy, go through, beta oxidation, TCA cycle, right, for complete oxidation. Okay, 40. How does muscle metabolize fatty acids during the fasting? I just said, right? During the fasting, fatty acids will be completely oxidized for ATP production in the muscle, right? Beta oxidation to acetyl-CoA, acetyl-CoA enter TCA cycle and complete oxidize and generate FAD, NAD, right? H, and then the reducing equivalent being transported to oxidative phosphorylation or respiratory chain, right, for ATP production. Okay, any questions from fatty acid oxidation you want me to talk about it? That's okay. You can just go back, even you're thinking about, I still have some question from lipogenesis. That's okay, right? Any question? Yes. Sorry? 37? Okay. 37 is more or less energy produced from unsaturated fatty acid oxidation compared to saturated fatty acids oxidation. More or less. Less, right? Because of a double bond present, so double bond cannot be break down, so you're less. Okay. Any other questions? Yeah. It's not on the study guide? Really? Oh, I'm sorry. Reverse transport, cholesterol transport, right? Okay, so yes, I think it is very important, should be included, right? I think I probably will go through the lipoprotein, all the question. I think it is probably most complicated, right? Okay, so let's see. Yeah. I saw you have a question, right? 33. Okay, so what is the sequence of reactions that occur during fat oxidation? Beta oxidation, right? Let's see. So I think what do you need to know, again, I mean, you don't need to remember the enzyme, reaction, substrate stuff, but again, it's also doing cycles, right, for beta oxidation. So in each cycle, you cleave to release one 2-carbon unit, which is acetyl-CoA, right? And also you need to remember how many FAD, H or NADH is generated, and that can help you to calculate the ATPs, from like the one molecules of fatty acid oxidation or something like this, right? And you also need to connect the beta oxidation with the transport of fatty acyl-CoA from cytosol to mitochondria. Beta oxidation occurs in mitochondria, right? And also connect this beta oxidation products like acetyl-CoA to TCA cycle, right, for complete oxidation. So I think this is that I would like you guys to know, but not specifically, which enzyme catalyzes which reaction or something like this, right? The general question. Okay. Anything else? Okay, ketone body synthesis. 43. Ketones are produced from what metabolic substrate? Acetyl-CoA, right? And how is this substrate produced? Yes, beta oxidation, and right. So basically from the fatty acid oxidation, right? Okay. I guess number 44. So liver mitochondria contain which key enzyme that play critical role in ketone body synthesis? Actually, 2 enzymes. I think I asking you to remember the common pathways, right, for both ketogenesis and cholesterol synthesis, and 2 enzymes that involved in the ketone body synthesis, HMG-CoA synthetase and HMG-CoA lyase, and which one is found in the cytosol or other cells and is involved cholesterol synthesis? Which one? The HMG-CoA synthetase, right? I think we have a slide. Okay, so this one. So this is the enzyme that involve both pathways, right? Cholesterol synthesis and also ketogenesis. 45. What is the principal driver of ketone body synthesis? What is the main driver? Yes, free fatty acids. Fasting condition, free fatty acids. Free fatty acid levels increase during fasting in the circulation is the main driver of ketogenesis. And 47. So what are the key regulators of ketone body synthesis? Let me see. If you still remember, we talk about it like 3 key regulators. Right? Regulation of ketogenesis is like the levels of fatty acids or lipolysis rate, right? If you have a higher rate of lipolysis, you should have more fatty acids be released. So circulating fatty acids level is the main drivers of ketogenesis, is also one of the regulators, right? And in addition to fatty acids levels in circulation, the CPT1 gateway, CPT1 activity is also the key, right? That can control transporter of fatty acyl-CoA from cytosol into mitochondria for beta oxidation. And lastly, the acetyl-CoA levels, right, also control the synthesis of ketone body, which makes sense, right? It's the acetyl-CoA substrate. So we have more substrate, and we should have a more ketone body can be synthesized. So 3 critical steps that can control the ketogenesis. Again, fatty acids level in the circulation is the main driver, right? Main regulator. Okay, 7. Oh, it's 47 talk about it. Okay, so any other questions you wanted me to talk about? Okay. All right. So for cholesterol synthesis, I guess you just need to know, what is a key enzyme, what is a substrate, or starting molecules for cholesterol synthesis. Acetyl-CoA, right? Cholesterol is synthesized from acetyl-CoA, and key enzymes HMG-CoA reductase, and in terms of regulation of cholesterol, I guess the cholesterol levels and methionine levels, substrate regulation, right, are the key regulators. And also in terms of the mechanism, how cholesterol or methionine regulates the cholesterol synthesis, basically cholesterol, HMG-CoA reductase, is through the transcription factor, SIBP, right? So this is something that I think you wanted to know. Okay, so the last part is the lipogenesis. The lipoprotein, right? Are you guys okay? You wanted to take a few minute break, or you're okay, right? Okay, so let's just finish. So lipoprotein. So what are the main 4 major group of lipoproteins? Let's see. Chylomicron, so chylomicron, VLDL, LDL, HDL, right? This are 4 main lipoproteins that I want you guys to remember. And which apoproteins is associated with each of this? For example, apo B48 is specific or unique to chylomicron, right? Because it's synthesized in the small intestine. And VLDL, I'm sorry, got it too. Okay, so and VLDL, apo B100 is specific for VLDL, right? Because it's synthesized in the liver, and chylomicron synthesized in the small intestine, and VLDL synthesized in the liver. And also, I wanted you to know that ApoC2 is very important apoprotein, right? What is the function of ApoC2? Is to facilitate LPL activity, right? So without ApoC2, LPL cannot be functional. So, and also, you guys wanted to know what happened, or what is the consequences of the ApoC2 deficiency, right? And also the ApoE and ApoB100, these 2 apoproteins are very important for LDL receptor functioning, right? So these are two apoproteins are required for binding of LDL to LDL receptor. And deficiency of these two apoprotein can also cause some problems, right? That LDL particularly affect LDL concentration or atherosclerosis. So 54. So what is the main lipid component of chylomicron, VLDL, LDL, HDL? So both chylomicron and VLDL are triglyceride-rich lipoprotein. That means they contain majority of the lipids in the core triglyceride, right? And LDL, HDL are the cholesterol-rich lipoprotein. 55. So which lipoproteins are associated or termed triglyceride-rich lipoprotein? Just described, right? Chylomicron and VLDL. And 56. What is the function of chylomicron? Where is chylomicron synthesized? In small intestine, right? Where the main function of chylomicron is to transport triglyceride from small intestine, which initially absorbed it from the diet, right? From small intestine to adipose tissue for storage. But I also want you to remember that chylomicron remnant is actually picked up by the liver, right? So the majority of the triglyceride in chylomicron being transported to adipose tissue, but some remaining triglyceride eventually being picked up by the liver. Okay, so what is the ACAT and CCAT function? So I think you probably wanted to know the specific slide that we have. I think we have a specific slide, right? Talking about this 4 different proteins, what are their functions and where they're located, something like this, right? Sorry, I didn't include it for some reason. So ACAT versus LCAT, I guess, yeah. So ACAT basically is catalyze the reacidification of free cholesterol to cholesterol esters, right? And the ACAT is located inside of cells, and LCAT it catalyzes the same reaction, reacidification, free cholesterol to cholesterol esters, but have a different location. So LCAT basically is present in the circulation, right, in the plasma. And we also mentioned the ABCA1 and the other one. The other one is the CEPT, right? The CEPT protein. So ABCA1 is the transporter of the lipids, particularly free cholesterol and phospholipids, and transport that actually located on the surface of a peripheral cells, right? And then can transport it cholesterol from. Let me just try it. Okay, so ABC1 again is a transporter, the transporter cholesterol free cholesterol and phospholipids, right, from peripheral cells and to beta HDL, and play a very important role in the process called reverse cholesterol transport, right? So the purpose of a reverse cholesterol transporter is to transport unnecessary or unneeded cholesterol from peripheral tissue back to the liver for the disposal, right? And the whole process is involved the HDL cycles between beta HDL, which is functioning as something like empty dump truck, right? And alpha HDL, so alpha HDL is a full dump truck. So this two version of the HDL kind of interconverted depend on during the reverse transport process, right? And the other protein I just mentioned, right? LCAT is in the plasma and catalyze reacidification free cholesterol to cholesterol esters, and CEPT is the protein that help the lipid exchange between the HDL and VLDL, LDL, right, and IDL. The lipid change basically is exchange basically between the cholesterol esters and triglyceride, right? So next slide probably give you better idea about the lipid exchange. So basically the HDL give the cholesterol esters to VLDL, but receive the triglyceride from VLDL, IDL, LDL, right? Okay, so so 59 will describe the process of making VLDL in the liver. I guess you probably want to go back to the this slide, right? So how VLDL is made, and you want to know where the fatty acids come from for making VLDL, right? I think we talk about the three sources of fatty acids, right, for triglyceride in VLDL. One is circulating fatty acids, can be fat conditioning, right? We have a lot high levels of fatty acids basically from diet, or also can be during fasting, right? We have also a lot of fatty acids being released from fat tissue. And the second is the synthesis within the liver during the fat conditioning, right? The lipogenesis, you can convert glucose or amino acids to fatty acids, and the third, basically during the disease condition, for example, diabetic condition, right? Usually they have high levels of fatty acids in the circulation, so that can also be picked up by the liver and for synthesis of VLDL. So VLDL secretion can be increased during the fat condition, and also during the fasting, right? The disease condition as well, like during in the diabetic condition. And once the VLDL is synthesized, right, and gets the necessary apoproteins, and then be transported to the adipose tissue for storage of lipids in the adipose tissue, again during the fat condition, right? But during the fasting condition, VLDL actually transported the triglyceride from the liver to muscle cell, right? I think we talk about this, right? We divide, the two different stage, fat condition or the fasting condition. The sources of fatty acids for VLDL synthesis can be different, and also the destination is also different, right? Fat condition destination would be adipose tissue, and the fate of the fatty acids would be stored in the triglyceride. And during the fasting condition, fatty acids you basically came from circulation, but initially from the fat tissue, right? Like mobilization. And VLDL will go to the muscle, and fatty acids in the VLDL being actually oxidized in the muscle during the fast condition. So there's two different physiological conditions, the different metabolism of VLDL. You guys probably also wanted to remember, right? And then the 60. So what is the main function of LDL, HDL, HDL cycle? Basically, it's a reverse cholesterol transport, right, process. So I think we already talked about it. And 62, how can free cholesterol pool in the cell be decreased? I think we have an important slide. I want you guys to remember. That is the synthesis, the regulation of intracellular cholesterol homeostasis, right? So, three pathways that adding free cholesterol to free cholesterol pool, and the other three pathways that can remove the unnecessary or unneeded cholesterol from the cell, right? Disposed, transported from peripheral tissue to the liver. So you probably want to just to review this slide. I think it's very important. And 64, basically, I think we talked about it already. And 65, interaction between VLDL, LDL, HDL, basically is lipid exchange exists, right? So where is the reaction occurs? In the plasma, right? The protein that catalyzes this lipid exchange is CEPT protein. So how the content of VLDL, HDL change? Basically, is lipids change, content change, right? For example, HDL gives triglyceride to the VLDL, LDL. Give the cholesterol ester, right, to the VLDL, HDL, and then receive the triglyceride from VLDL. So they become more triglyceride, to get more triglyceride and get rid of cholesterol esters. And the two enzymes that involved in this process, like CPT1 and LCAT. LCAT basically catalyzes reacidification, right? Okay. Yeah, I know what you talk about, and I probably this question pretty kind of general, right? So you thinking about a little bit more specific question, I guess. Is that what you're asking? Can't remember who you, right? Yeah, who asked, there's no reverse crystal transport questions? Oh, you, sorry. Talk about it. Okay, yeah, maybe I can rewrite some specific question and send to you guys, right? Okay. So the last part, the essential fatty acids. I think it's pretty simple, right? You guys just wanted to know the definition of essential fatty acids, why this linoleic acid or alpha-linolenic acid are the essential fatty acids, because they are cannot be synthesized in our body due to lacking the two important enzymes, right? Delta 12, delta 15 desaturases. So we must obtain these two fatty acids from the diet. So this essential fatty acids. And so how does the essential fatty acid, non-essential fatty acid metabolites differ? Basically, non-essential fatty acids, the main function of this category of fatty acids is for it is to oxidize and for energy, right? And the essential fatty acid basically is not usually is not utilized or metabolized for energy, but it's very important cellular component and also function as signaling molecules, regulate the some pathway activities. And so what does omega mean? And you guys know, right, just a numbering system, the first double bond location, if it has the omega-3 position, like this fatty acid called omega-3 fatty acids, if that is the omega-6 position, the first double bond, right? And the is basically the reference omega-6 fatty acids. So the fatty acid or essential fatty acids, omega-3 and 6 fatty acids, basically polyunsaturated fatty acid, right? Have more than one double bond, so multiple double bonds. Yeah, I guess the omega-3 fatty acids has other two family members, right? The DHA and EPA, and omega-6 members are arachidonic acid, arginic acid, and they have different functions. So omega-3 basically is considered anti-inflammatory, and omega-6 is the pro-inflammatory because of the precursors for the synthesis of mediators, right? So omega-6 fatty acids, particularly arachidonic acids, is the substrate for the pro-inflammatory biomediator, eicosanoid or leukotriene, etc. Right? And omega-3 fatty acids are usually used for synthesis of anti-inflammatory eicosanoid or biomediators. So they have a different role, right? I think pretty much that's all I thought is kind of important for you guys. Do you guys have any other questions? Okay. Yeah.
What did you say the function of the ACAT was?
ACAT?
On the license.
ACAT is located inside of cells, and the function of this enzyme is to catalyze acidification of free cholesterol to cholesterol ester, right? And LCAT is the similar enzyme that catalyze the same reaction, but have a different location. So LCAT basically is present in the plasma, right? Yeah. 48. Does ketone body require energy to enter mitochondria for oxidation? Yes or no? Yes, right? Remember the utilization of ketone bodies, one of the steps that has a key enzyme, succinyl-CoA transferase, catalyze the reaction, right? This reaction requires ATP, usually GTP, I guess, right? Requires ATP. So ketone body utilization does need energy, right? And liver cannot utilize ketone body because of lacking of enzyme, right? Guys, remember succinyl-CoA transferase, right? Okay. Any other questions? No? Okay. So yeah, I'll just try to remember, right? Some more question about the lipoprotein, right? Particularly reverse transporter. Yes. No, actually, the last practice question just, to help you guys get familiar with the format. We don't have specific, the practice question for each session. All right. Okay.